In this topic, we're going to have a look at bulk transport. So by the end of this topic, you should be able to answer the questions, what is bulk transport, what is endocytosis, and what's exocytosis? So in the previous topics, we've looked at how individual molecules or ions can cross membranes. Now, there are methods to transport large quantities of materials into or out of cells. You've got endocytosis, which involves the gulping of material, and then exocytosis, which involves the removal of material from cells. So it's quite easy to remember: endo coming in, exo exiting. So endocytosis takes two forms. You've got phagocytosis, which is known as cell eating, and pinocytosis, which is cell drinking. As mentioned earlier, endocytosis is the engulfing of material by the cell surface membrane or plasma membrane to form a small sac, which is called an endocytotic vacuole. This is also called a phagocytic vesicle or food vacuole. And this process is active and requires ATP. Examples are white blood cells and amoeba. Let's have a look at the different steps in phagocytosis. So phagocytosis is the bulk transport of solid material. The cell responds chemically to food or bacteria and it moves towards it. The cell surface proteins recognize and attach to the food or bacteria. Then you've got pseudopodia, which extend around the bacteria. So they look like arms, cytoplasmic arms. This involves microfilaments and ATP. Then the cell membrane invaginates and pinches off, which means it forms a cup-like depression in which the large particles are contained. This forms a phagocytic vesicle or vacuole. We also call this a phagosome. Then you have a lysosome, which contains digestive enzymes such as lysozyme. It fuses with this phagocytic vesicle or vacuole to form a phagolysosome. The bacteria then gets digested by the enzymes. And finally, the undigested debris is removed by exocytosis. Okay, let's have a look at pinocytosis. So pinocytosis is cell drinking, and it's similar to phagocytosis, except the vesicles are smaller. It's often used for the uptake of liquids. So you have pinocytic channels, which are formed at the cell surface by invagination of the membrane. Fluid from the environment moves down the channel, and the end of the channel pinches off, forming small pinocytic vesicles. If the vesicles are very small, we call them micropinocytic vesicles. And these vesicles can move around the cell, for example, taking water to parts that are needed, where the water is needed. Examples include the uptake of fat droplets by micropinocytosis at the base of villi and small intestines, and reabsorption of amino acids in kidneys. This shows you how materials exchange between the capillary and the muscle by pinocytosis. Notice the little pinocytic vesicles. And here you can see how material is transported across the endothelium of the capillary in those little tiny pinocytic vesicles. So far we've discussed phagocytosis and pinocytosis, and there's something else called receptor-mediated endocytosis. So here you can see the different forms of endocytosis. In phagocytosis, it's like cell eating, pinocytosis, cell drinking. And then in receptor-mediated and endocytosis, you've got receptors binding to molecules and the plasma membrane invaginating to form vesicles containing both the receptors and the molecules. And there is a protein called clathrin which helps to form the vesicles. Okay, let's move on to exocytosis. Exocytosis is the removal or secretion of substances from a cell. Vesicles bud off the Golgi apparatus to form secretory vesicles. These fuse with the cell surface membrane and their contents are expelled into the medium outside. So examples include enzymes being released from the pancreas, endocrine glands, and then cell wall deposits. Removal of undigested waste in phagocytes, as we've discussed, pancreatic enzymes, and then the release of hormones, for example, insulin. And here's quite a nice picture of exocytosis, where you can see the contents being released out of the cell. In this case, a secretory granule is being released out of the cell. And finally, this is quite a nice summary of the events that we've just discussed. Can you see pinocytosis, exocytosis, phagocytosis? So remember, pinocytosis is like cell drinking. Look at the events with pinocytosis. Exocytosis, notice how it's relating to the Golgi apparatus and how the vesicle is budding off from the Golgi apparatus. And then phagocytosis, notice how if it's like a bacterium, the bacterium will be engulfed. Then you'll have a lysosome coming from the Golgi apparatus, a vesicle from the Golgi apparatus, fusing with this phagocytic vesicle or vacuole, and that's how the bacteria gets digested. Okay, in summary, we've looked at the three mechanisms of bulk transport. Endocytosis, which is the engulfing. So we've looked at phagocytosis, and remember these keywords: pseudopodia, pinocytosis, which is cell drinking, which is, it involves pinocytic vesicles, and then exocytosis, which is the removal of materials, and these involve secretory vesicles. And that concludes our lesson. The end.